A neuron is a nerve cell that is responsible for sending an electrical signal to the skeletal muscle cell, since the skeletal muscle can only contract when they get a signal from the neuron. The neuron has two parts, a dendrite and an axon. The axon is the part of the neuron that interacts with the skeletal muscle. An electrical signal called an action potential is sent through the neuron on the cell membrane. Since there is a gap between the axon and the skeletal muscle, it needs a chemical signal that will stimulate another electrical signal on the skeletal muscle, which occurs in the synapse between the nerve and muscle. Once this chemical signal has occurred, the action potential will go down the skeletal muscle, causing it to contract. Let us take a closer look at the chemical signal that is needed to produce another electrical signal. This is a closer look at where the axon and the skeletal muscle will interact. This is the motor end plate of the skeletal muscle. This is the synaptic knob of the axon. These are the synaptic vesicles within the synaptic knob. The synaptic vesicles are the cavities that hold the acetylcholine, which will be responsible for the chemical signal throughout the process. There are calcium ions on each side of the axon of the neuron. There are sodium ions between the synaptic knob and the motor end plate, also referred to as the synaptic cleft. Just below the motor end plate are potassium ions. The action potential that follows down the neuron's cell membrane passes the calcium ions where there are calcium ion channels. The action potential sent through the neuron opens the calcium ion channels. When these calcium ion channels open, the calcium floods through the channels and makes its way down the axon to the synaptic knob. The calcium ions cause the synaptic vesicles to move to the cell membrane and then fuse with the membrane, causing exocytosis to begin, which will release the acetylcholine into the synaptic cleft. The motor end plate is aligned with acetylcholine receptors. Once the acetylcholine is within the synaptic cleft, it will find its way to the receptors and stay on them. Once the acetylcholine is on the acetylcholine receptors, it will open sodium ion channels that are also on the motor end plate for the sodium ions that are within the synaptic cleft. Then the sodium ions are on the other side of the motor end plate with the potassium ions. What has happened with the sodium ions is called depolarization, where the charges of each end has flipped with the sodium ions and potassium ions on the same side of the cell membrane of the skeletal muscle is the sarcolemma. This depolarization from the sodium ions and the potassium ions being on the same side of the sarcolemma causes a big enough depolarization to hit threshold, which will cause a new action potential on the muscle cell membrane. Let's take a closer look at the sarcolemma and the skeletal muscle structure. In the skeletal muscle, there are fascicles, and between each fascicle are tubes called T-tubules. Between the fascicles are mitochondria that are attached to them as well. On the outside of the fascicles is the sarcoplasmic reticulum. The inside of the fascicles are made up of myofibers, the myofibers are made up of myofibrils, and the myofibrils are made up of myofibrils. Let's take a closer look at the sarcoplasmic reticulum. The new action potential from the chemical signal was left off going down the sarcolemma of the skeletal muscle. The action potential continues to go down the sarcolemma until it comes to a T-tubule, which will fall down through the terminal cisternae of the sarcoplasmic reticulum. The terminal cisternae of the sarcoplasmic reticulum is filled with calcium ions. There are so many calcium ions that the cell has another molecule called calsequestrin to store the calcium ions in the terminal cisternae. When the action potential passes through the terminal cisternae, it opens calcium ion channels, allowing the calcium ions to flood out the terminal cisternae. Now let's go to the second part of the process within the myofilaments. The myofilaments are made up of individual sarcomeres. Each sarcomere is separated by Z-discs. The A band of the sarcomere runs the length of the thick filaments. The I bands of the sarcomere run along the part between each thick filament. The H zone runs along the middle of the sarcomere and only where the thick filaments are. The thick filaments are made of myosin. Those myosin have two myosin heads. The thin filaments are made up of two strands of actin molecules wrapped together, with each one having an active site. It's also made up of tropomyosin, which is two rods twisted around the actin and covers the actin active sites. The last thing it's made up of is troponin, that is attached to the tropomyosin. An ATP molecule attaches itself to the myosin head, giving it the energy to go into the myosin's high energy state. The calcium ions from the sarcoplasmic reticulum attaches to the troponin, causing the troponin to pull the tropomyosin off the active sites of the actin. Now that the active sites are exposed, the myosin will attach to the active sites of the actin in its high energy state. Once the myosin has attached to the actin, the energy from the ATP molecule has been used, and what is left is an ADP molecule and a phosphate molecule, and when the myosin molecule attaches to the actin, the phosphate group also falls off, leaving the ADP molecule only. Now the myosin head will go to the low energy state, causing the thin filament to move with it. The ADP will also break off while the myosin is still attached to the actin. Now that the myosin head is in low energy state, another ATP molecule can attach and restart the whole process.